Our question was how will using different enzymes affect DNA extraction of a pineapple?
We hypothesized that the bromelain would not work, but the papaya enzymes would. This is because bromelain is already present in pineapple, whereas papaya is not.
Our materials included a test tube rack, 3 test tubes, a can of pineapple, a buffer solution made of soap, water, salt, and enzymes, of which we used bromelain and papaya, cold ethanol, safety glasses, a water source, cheesecloth, a funnel, and a graduated cylinder. We began our experiment by putting crushed pineapple into a bag with the buffer solution containing the bromelain enzymes. After mashing the pineapple mixture for around 1 minute, we poured it into a funnel that was covered in cheesecloth. This process was to break up the cells and their membranes. After straining the majority of the liquid into a test tube, we added cold ethanol. This was to separate the DNA from the rest of the molecules. However, our results were not as expected. During this first test, we used the pineapple enzyme bromelain. Since the enzyme is already present within pineapple, it reacted differently than it should have. The solution began to bubble instead of separating and showing the DNA like it normally does. We still don't really know why this happened. One theory is that the solution was so overloaded with bromelain enzymes that the enzymes started breaking cells apart. This is what created the oxygen bubbles. But like I said, this is just a theory. On the next day, we started the second half of our experiment. We used the same process as the first half. All we did is change the enzyme from bromelain to papaya enzymes. From the beginning of the second round of testing, we saw slight changes. Although the solution was still bubbling when we added the cold ethanol, we found that with the papaya enzymes, the DNA was beginning to precipitate more than the other solution. Although we could see the DNA precipitating, we could not collect it. This is because it was not sticky enough and in not high enough quantities. Our 3 tests throughout the day varied quite a bit. The first test had a decent amount of DNA. The second test had a very large amount. And the third test ended very similar to the first. Why did all of this happen? Why did the solution start to bubble? Why did the papaya enzymes work better than the bromelain? These questions are still unknowns. But what we do know is that neither of these methods are good for extracting pineapple DNA. The rest will remain a mystery.